Welcome everyone to another iPlant Focus Forum webinar. Today we have Jorge Duitama and Juan Fernando de la Hoz from CIAT in Cali, Colombia joining us. My name is Martha Naro. I'm with iPlant and I'm in Tucson, Arizona. So I will now let Jorge and Juan take it away.
Okay, thanks, Martha. Good morning, everybody. So, welcome to this seminar on the work in which we will present part of the work that we have done on our software tool called NGSEP, which for analysis of high throughput sequencing rates. The way this webinar will be organized will be the following. So I will start with a few slides to let you know a little bit of the background of this project. Then Juan Fernando will make the practical part of the webinar, showing us how we integrated NGSEP into the iPlant discovery environment and how can we use it. And then I will finish and we'll take a few questions, and I will finish with a few more slides on the experiences of our users. So let's just go ahead and start. Okay, no, sorry. Before that, so should you have any questions, just let's to make it organized. You can use the chat. We will have a few stops in the way, and then we will read the question and answer the question. So again, I will start saying a few words about what is CIAT for people that is not familiar. So CIAT stands is the Spanish acronym for International Center for Tropical Agriculture. We are located in Cali, Colombia. Here is a picture of our campus. If you have some time, come and visit. The weather is great, so you are very welcome. You have a chance. But we are because we are at the international center. We also have regional offices in Nairobi mainly for bean breeding, and in Hanoi for cassava in Asia. So we belong to a large consortium called the CGIAR. The goal of the CGIAR is on here in the right. You see the main goals of this large consortium. We are 15 centers located mostly in tropical countries of the world, and the objectives are to reduce rural poverty, increase food security, improve human health, and to achieve all these three objectives, ensuring also sustainable management of natural resources. So as you can imagine, these are pretty big objectives. It's a very difficult task to accomplish, and we need an interdisciplinary team, people with many different skills to do that. In particular, a lot of the work that CIAT has done in the latest 50 years have been breeding to develop improved varieties, most of the time with conventional breeding. And the objective of the genomics work, and definitely from the bioinformatics work here at CIAT, is to support breeders, support the good information with genomic tools, with whatever we can take from the genomic component, or even from transcriptomics or genomics components of the plants, of how the plants behave, to help breeders to take informed decisions and to reduce costs, reduce times, and accelerate the development of improved varieties to alleviate hunger and poverty problems. So for that reason, we are pretty well aware of this that that many people call the genomic revolution. So what you see on the right, this chart that you see on the right is pretty much a commonplace. Probably you have here almost every single genomic talk. But that doesn't mean that it's not amazing. So what you see here is how genomics and sequencing a piece of DNA, sequencing a molecular DNA of any organism, have gone down from an effort of millions of dollars to an effort of, let's say, even less than $1,000. And the reason for that is the development of these high throughput sequencing machines. So what this means is that in current numbers, if you have, let's say, $10,000 that you want to spend in a sequencing project, you can do a good job making whole genome resequencing of 4 human samples, But in the case of CIAT, we have 4 mandatories, so rice, cassava, bean, and forages. So you, for example, can sequence 13 samples of the whole genome sequence of 13 samples of cassava, 17 samples of beans, 17 varieties, or 19 varieties of rice. And as you can see, that's more or less a linear function of the genome size. You can do a little bit of a better job with beans and rice because they are naturally inbred, so they have less heterozygosity, and that helps. But then one of the big challenges of every institution doing this kind of work, and in particular of CIAT, is that if you just spend that money, you spend your $10,000, and what you have back is not what you would like to have, which is perhaps for each DNA molecule a sequence that will tell you the chain of nucleotides that will represent that molecule that you are investigating. But what you will have is a bunch of millions of these small reads. And then in a file called FASTQ, which is more or less difficult to process and to read, and definitely you will, by hand, you will not be able to do anything smart with that kind of file. And that's where bioinformatics come to help. So what you will need to do is software to help you to process that data, this huge data, and extract the information that you need from the experiment. So, but before going to the details on how that works, let's assume for a second that you already have, that we have a magical machine that is able to read DNA from left to right, and that you do that for, let's say, the same chromosome or for a homologous sequence in a couple of two samples, or it can be on more samples. So what this cartoon, to explain you better, so to explain you better the type, to go towards what NGSEP does, this is a cartoon that tries to summarize the types of genomic variability that you can find between two samples. So many people is familiar with the concept of SNP. A SNP is like the smallest piece of variation that you can have, and it's basically a stretch of DNA in which you have in the two samples exactly the same sequence, except for one particular base, but when you have one nucleotide in one sample and a different nucleotide in another sample. You can have also a small presence absence, so nucleotides that are present in one sample and not present in another one, so small indels. A particular case of small indels are the tandem repeats. So tandem repeats are small sequences that are repeated many times, and the variation comes from the number of copies that you have of each sample. So these are small events. In a random process, they will have little chance to, a smaller chance to have an effect on the genome, but depending on where they are, they could actually have a large effect on the genome. But then you also have variation that is larger, and for that reason they call it structural variation. So you can have larger regions of DNA that are present in one sample and absent in another one, so large indels. You can have kilobase pairs or megabase pairs with presence absence variation. You can have pieces that are in only one copy in one sample and more than one copy in another sample. So that's why that's called duplication, or more general copy number variation. Or you can have pieces that are exactly the same sequence, but going in one direction in one sample and going in the opposite direction in another sample. For that, you need to remember that DNA molecules have you can define the sense of direction as going from 5' to 3' normally. And also you can have pieces of DNA that we call active. In particular, transposons are a good example of molecules that are more or less actively moving around the genome, and you can call translocation events when you detect events like this. Okay, so NGSEP started basically to give you basically an idea of how analysis pipeline works, and also to tell you a bit of story on how NGSEP started. So the process that you do when you have the reads and you have a reference genome, which is more and more the case for more species as sequencing projects are getting to an end, what you do is you align the reads, which means trying to infer for each possible read where in the genome it comes from. And then you sort this read by reference coordinates, and then you have what you need to, and then it starts making, with this process, you start making a little bit more sense of the data. And then you do a process in which you go base pair by base pair in the genome, and you try to infer the most likely genotype in your sample. That's core-SNP detection. Again, that's how NGSEP work. I had to do exactly this for RNA when I was a PhD student. I needed to do this for RNA-seq data in cancer tumor. There were not too much software available at that time, and we were in computer science. We wanted to develop algorithms, so I ended up writing a SNP code for this particular case, which later grew up to be the application that is right now. So I will not go into the details of how the algorithm works, because for that we'll need a whole complete new seminar. But I wanted to give you like a first idea of how this works. So you go with them locus by locus, and if you use information, the base calls that are present in that particular locus, and if you know that that every single base comes with an error probability that the Illumina sequencer gives you like a likelihood of a probability of error reading that base, you can infer the random genotype. So you can assume that the genotype is a random variable, the genotype that you want to predict which of the possible genotypes is the genotype present in that particular locus. And most of the software tools, not only NGSEP, but also some tools, the ATK, the other software tools, most of the other software tools that perform SNP detection. The way to formalize the problem is as using the Bayes theorem as a Bayesian problem, in which you calculate the genotype with the most likely posterior probability, calculating condition, setting up a model in which you set the prior probability and a condition probability of observing the reads given each particular genotype. And that's in general terms how it solves the problem. The formulas are right there, or they are also in the papers that we published before. And that's for, let's say, SNPs or even small events. For structural events, there is a large literature in bioinformatics on several different algorithms. Most of the algorithms follow one of these two strategies, which are called read depth or read pair. In read pair, what you the main assumption is that for each base pair or each locus on the genome is on the sample is sequenced at has a random chance of being sequenced, so the distribution of the reads should be uniform on the sample. So when you align the reads to the reference, if you detect loci that have significantly less coverage or significantly higher coverage than the average, then you can infer that there is either a division or a duplication event, and in general that there is copy number variation in that locus. And then the way read pair works is if you do parent sequencing, you know that the two ends of a pair should be close to each other, should be facing each other. There is an expected distribution of how each pair should be aligning to the reference. So when you observe that the two ends are aligning too close or too far, or they are aligning in the wrong direction, then you can call them either an insertion or a deletion or an inversion, respectively. So what we did with NGSEP, so what we noticed and what people in human genetics notice is that the best thing you can do is to combine all this together, to combine all these different approaches together, and then to achieve, to try to maximize the number of events that you can get. There are events, I think it's nearly impossible to call everything from short reads because of the nature of the reads, and to have a complete perfect representation of your sample, of the DNA sample, but you can get very close combining all these efforts. And that's what we did with NGSEP. We wanted a single tool. So the problem, one of the problems that we observed is that for these algorithms, there are a lot of software tools. They are different. They have different formats. We even found quality problems. It's really a pain for biologists to try to put all this together and try to analyze them as a single thing. So the approach that we took was to try to develop a software tool that will integrate all this in a, let's say, easy to use fashion. That can either be our graphical interface or with a few clicks on a graphical interface, or it can be also a single command or a small set of commands in the command line environment. So what we did was we finished this, the software, we finished the graphical interface, and that's why we call NGSEP the software in the beginning. NGSEP stands for Next Generation Sequencing Eclipse plugin. So our graphical interface, we did it as a plugin of the Eclipse, which is a pretty well-known environment in the software development world. We use Eclipse because it facilitates a lot of things. So we have a file manager that help users to see their files, see the files corresponding to the different samples, organize them, call functionalities based on the file manager. We also have a processes manager to make different processes in parallel. So it helps us a lot. And then we build the screens on top of it that again allow the user to find variants or to go to the different functionalities in an easy to use platform. That's particularly good for people who is learning how to process reads. We start with next-gen sequencing, and also for people who have limited skills in command line and programming skills. So so we made the software available. It's perfectly open source. It has been like that, and hopefully it will continue to be like that. And we publish it. We have a publication about a couple of years ago, and you can see the main features that we had at that time and the benchmark that we did against the comparisons that we did with other tools. So that doesn't mean that if you are not experienced on command line environment, you can use our graphical interface. But if you are experienced, if you feel comfortable with the command line, and if you have a lot of experience in informatics, and if that's actually most of the way we work most of the time here at CIAT. We also have everything available in a command line environment. So what we did was to pack everything in a single jar file, and you can call it as a normal Java application. And actually, you can do it in any operating system. You can do it in Windows, in Mac, or in Linux. So I'll explain. So this is the list of the main functionalities as commands of the tool. So basically, the main command and the main functionality is find variants, and that's the functionality that integrates all these algorithms I was telling you about. Then we have a process to merge variants from different samples. Then when you have a VCF, which is the format that was developed in the Human 1000 Genomes project to represent genomic variation with qualities and with coverage and different aspects of the genetic codes, you can annotate that to predict the functional consequences of your variation related to the annotation of the genome, and then you can filter. You have several options for filtering and to export or convert the VCF to several different formats, or to GWAS, or to structure analysis, or for a lot of downstream analysis you can make. And we have further commands. We have a growing number of commands. My suggestion will be if you are interested, to go through our tutorials, through our material for learning material, and then you can get to see what a lot of the things that you can do with NGSEP, which include again also statistics that can guide you through statistics not only on the sequencing process, but also statistics that will make sense from the population genomics. And this year, actually with a lot of the help from Fernando, we have increased and we have expanded the number of ways in which you can use NGSEP. So, so the way from the architecture, let's say, point of view, so for architecture, this is how it's organized. So we have our core functions. So if you download from our webpage NGS2app version .jar, what you will have access to the core functions written in Java. And then I wanted to emphasize at this point that no matter from where you call NGSEP, you are calling exactly the same code. No matter if you do it from iPlan, Galaxy, no matter from what you're doing, is exactly the same code. In the core functions, we have also in this jar, we have the command line interface, the pin interface. That's what you interact with. You use it in command line, or if you use the Eclipse plugin, get access directly to the functions. And then, if you are familiar with Galaxy, if you work with the Galaxy environment, you want to connect NGSEP with other tools using Galaxy, and you have that you have Galaxy already implemented in your servers, you can use make the XML wrappers that facilitate the process for you guys, and with documentation to help you to facilitate the process of integrating NGSEP into Galaxy. We are working in a collaboration in the 3000 Rise Genome project. That a lot of the analysis of that is done with the help of a software company called DNA Nexus, that offers software as a service, bioinformatics software as a service in the cloud. So the variant detector is also available. They made it available, help them to make it available in their solution. And then, but then the most important and the interface that we are promoting today is the iPlant integration. So iPlant, as you know, is a large collaborative effort to have access to a lot of hardware resources. And not only just hardware resources, but there are a lot of software tools that are implemented into this environment. And for us, I think was really interesting. So we started early this year. We started with the integration of with iPlant through the development of iPlant applications. And that's what we'll show today in the demo. Just to make it easier to go through the pipeline that Juan will present, this is a schematic of our pipeline to go from, of the pipeline that we are using to go from route reads to BCF, combining variation from many individuals. So we do, again, read alignment and sorting. For that, we don't have any specific, we haven't developed anything new. So there are a lot of software tools that are available for that. Some of them implemented in iPlant. So we tend to use here both the two, but people are very familiar also with VWA, or there are a lot of software tools that are available for that. It doesn't matter what you use. If it produces a proper MAM file, and you can sort it either with some tools or with Picard, or with any other tool by reference coordinates, that's the input that you need to operate NGSEP. And then the main pipeline includes the variance detector, variance detection step, where you infer which are the variants of each particular sample. Then you put the samples together in a variance merge that only produces a catalog. It still doesn't produce the final decision, but only produces a catalog that has all the sites in the genome that are interesting, that at least one of your samples says that can have variation. Then you go again sample by sample, go back to the variance detector to do genotyping, to genotype these particular sites, to focus on these particular sites and infer the most likely genotype for these sites. So you get at the end a VCF file for each sample, but with more complete information. And finally, you merge together these VCF files that you produce at this step to produce the final VCF. That's the pipeline as we have it organized right here. So in blue, you have the what you need to do sample by sample. So for each of the samples that you need to analyze, and in green you have the processes that produce one single file and makes one single file per population. So I will stop at this point, and before giving the word to Juan, I will see if you guys have questions at this moment. Let me stop sharing. Okay, so let's see what we have in the chat right now. So definitely we have a copy of the slides. Let's go down. So Alex, in the absence of a reference genome, could you use a related species? The answer is yes. So the reference genome is basically, so what people use are, I mean, first of all, there are approaches for do de novo assembly, or for do de novo, in particular for if you have GDS, or if you have rat sequencing, there are options, and there are a few software tools that can help you if you don't have a reference genome. We right now don't have access directly for completely de novo analysis. But my personal suggestion, if you don't have a reference genome, is to build at least a draft that you can use to align rates. If you have a related species, you can use it. The problem is that because you are far from your, the farther you get from your species, the less reads you will be able to align. That's kind of the problem, or you have to be more sensitive, allowing a few more errors in the alignment. But definitely you can do the job. If you have if you can somehow make even a draft reference genome, even if it has a large number of scaffolds, you can align your reads. But if you have something that you can use to align your reads, then you can work with the software. This should all be, and then Alex asks us if this should all be possible with circular genomes like mtDNA or chloroplast. I'm thinking that will be useful. So yes, I mean, from the usage of the software, the more complete your reference, the better. No, so if you have normally, for example, for rice, we include the mitochondria, the chloroplast, and then we can see if we have if the reads coming from mitochondrial DNA. We have definitely including that's something we can include. So we have another question from AGI. Is there a step of read alignment to have more confident call of read realignment to have more confident call of variation? JTK has it. So our approach. So JTK made has a module called Intel Realignment and quality score recalibration. In particular, they did a lot of work to make a quality score recalibration. What we did was to take two alternative approach. No, for the Intel Realigner, the difference with JTK is that we don't have it as a separate step, but we have it within the software. We have it built in the Intel Realigner in the software. So you don't notice it, but the Intel Realigner is happening when you process the reads. And we have done several improvements. Actually, in the latest version, we made a lot of improvements to make better Intel Realignment from the reads. So you don't need it as a separate step, and the advantage of that is that you don't need to produce a new one file. So we are building the Intel Realignment within the variance detector is good for users because it reduces the amount of space that you need to process the sample. And then for quality score calibration, what we replace it with a parameter in which we control how the maximum number of the maximum base quality score, basically to control to reduce the error produced by reads by base pairs are not called right, but with a high quality score. Of course, the good news also is that Illumina is getting better and better. It's producing better reads with better quality score, so that also helps. In the case of low coverage, so Carlos asked us if in the case of low coverage sequencing GBS, is it possible some sort of imputation? So right now, yes. One of the functionalities that we released recently, early this year, was imputation for inbred samples. So if you have rice, if your population is, let's say, nearly double haploids, so such as rice, such as beans, you can use our imputation module, and you will have a good quality of wheat. What we did was to re-implement a hidden Markov model that was already published. And this will provide you an imputation of very high quality. For outbreeds, so normal diploid samples, I will recommend, there are several software tools that are available. We were using the tool that we were using because it's more comfortable, because it receives DCF files, is Beagle. So we have to do some imputation with Beagle here. Again, there are other software tools that are a little bit more difficult because you need to transfer. We have, for example, the converter to phase, but phase is very slow. So you will need to do a bit of technical work, but there are more tools that can help you with sequencing. If you have inbred samples, then you can use our imputation module. Alex, lots of ways to generate SAMBAN. So an integrated de novo assembly probably will not be desirable. Okay. So Alex is telling us, yeah, I mean, again, it depends, depending on how far are you from the reference. You definitely don't have a good reference. You would like to do a no assembly, or if your population is very far from something that you can map. Again, it depends on if you have a reference or not. But again, I still think is the process is more efficient, and you get better quality SNPs if you use something you use a reference like anything that you can use to organize your bits. So Lia is telling us that there is an impute application in the discovery environment for the implementation. Definitely, I mean, a lot of these tools are implemented in iPlant. The problem with some of the tools is that they still, again, one of the problems, but it's a problem with the tool definitely, not of iPlant, is that they don't receive BCF files or they receive their own file and you have to build your own converter. In that regard, if there is a tool that is interesting and you would like to export BCF to that particular to a particular format that will be interesting, you can always write a email and we could add new formats to the converter at some point. That will be something we could do to include that in NGSEP. What else? Do we have more questions at this point?
A tutorial sample.
Is there a tutorial sample available to test the app? Yes. I think Juan will be the best person to explain the tutorials. But definitely, we have done a lot of work this year to improve the tutorials, especially for the command line, because is the phase that will need more training. And yeah, if you go, everything is available in our web page. We are even we have the project of improve our web page, not something within SourceForge, but our own web page. So definitely there is a lot of material that is already available, and hopefully there will be more to come in the next few months.
Okay. Yeah, and that's also the samples I'm going to show during the tutorial.
So yep, Juan, please come back to the word.
So welcome again. My name is Juan Fernando. I am a biologist and I am working at CIAT since 1.5 years. So now I am going to show you how to work in NGSEP in the discovery environment.
Let me interrupt you for a second. So the web page, the link to our web page is actually in the presentation. If you look at if there was a slide in so if you look through the slides, you will see the URL of the web page. You can make it available at any time. Yeah, sorry.
So can you all see the discovery environment?
Yes.
Perfect. So this is the discovery environment for the ones who don't know it well. Here are 3 buttons that allows you to open windows for the data, for your apps, or for the analysis that you're performing. In the data window, you can find here in the community data folder, in the iPlant collaborative directory, the example data directory, and down here, there are the samples, the test data for NGSEP. So here you will find some files that will help you to move around to use all the functionalities available in NGSEP. And the ones that we're going to use today are here in the directory webinar. Also, if you open the apps window, you will find all the functionalities of NGSEP here as iPlant apps. As Jorge told you, the main one is this, the variance detector. So that's the one we're going to use today. Okay, to start, I will show you the files that we're going to work with today. They are common yeast samples, Saccharomyces cerevisiae. We are going to use common yeast because it is a small eukaryote and it allows us to use all the functionalities of NGSEP. We are going to use these 2 strains, ER7A and CBS4C, because, well, they were sequenced some years ago with Illumina technology and they are pair-end, so we have two read files, which were then aligned to the reference genome that you can find here in FASTA format using Bowtie2. So this produced these 2 alignment files, and we sorted them afterwards using Picard. So that was the pipeline to get to this point. And here you can, if you want to start from the beginning, here are the read files. And if you're going to start from where we are going to start today, then here are the BAM files for the pipeline. All right, so now that we have the reads coming from the sequencing experiment of our samples aligned to the reference genome, we can see what are the differences between these samples and the reference genome. For that, we are going to use the variance detector. So when we open the variance detector, we can put here an informative name for this test. So this is going to be the name of the sample. And the main arguments, there are only four mandatory arguments. There are I have to load here the sample file, then a name for the output, the reference genome in FASTA format, and the name of my sample. And that's it. I can launch the analysis with default parameters, and then it will start executing. Up here in this icon, I can see that it was submitted, and once it starts running, I will see it running. Okay, for the other sample, I will do the same. So just modify the name of the analysis. Load the sample file. And the reference genome. And that's it. I can launch the analysis with default parameters, and it will start looking for all the variation present in my samples when comparing them to the reference genome. Now, I will try to explain some of the parameters most commonly used in NGSEP. So first of all are the four that are mandatory, but then you also have, you can also enter known variants from your samples or for the population, like if you want to genotype variants in the samples that you're analyzing instead of discovering new variants. You can also trim the reads in both ends if you know that the quality at any end is not optimal. And this is the list of the analysis that NGSEP will perform. So you can choose to skip some of this analysis. In this case, for example, you will skip all the structural variant analysis that Jorge already explained. And if you check here, you will skip the single nucleotide variants analysis and the small indels. Then the options for the small nucleotide, single nucleotide variant analysis are here. The most important ones are the heterozygosity rate, just to tell NGSEP how often do you expect a heterozygote to come in your sample. The genotype quality score, the minimum genotype quality score for a variant to be output. The maximum base quality score, that was what Jorge explained about the quality scores coming from Illumina. We suggest 30, but you can modify this too. And here you can set a maximum number of alignments per start position. This is our way of removing possible duplicate reads from PCR artifacts during the sequencing process. The ploidy of the sample you can also modify. And I have to warn you here, if you're working with reads aligned with BWA, you must check this button too. Okay, and the structural variant options include the bin size of the window to slide around the genome to see for differences. You can choose some algorithms for CNV detection. We have available CNVinator and event-wise testing, or both if you want. And also a minimum structural variant quality, a threshold for outputting to output the variants. Yes, and here you can also set the overlap between repetitive regions and CNVs detected by NGSEP. Okay, those are the main parameters. Once we can see here that our samples, our processes are running, So they are going to take around 20 or 40 minutes, so I won't wait until they are finished.
Okay. So, let's stop here for a minute to see if you guys have any questions. I see a question in the chat. So AGI is asking us if if the discovery environment default parameter is the same as in the command line default. The answer is no. We did it. Most of the default parameters are consistent. They should be perfectly consistent with the Eclipse plugin, because in both cases, because they are set for people to start working with this, we set up more or less conservative parameters. In the command line, we did something a little bit different, because we assume that people is more experienced. We adjust the parameters in a way where you maximize sensitivity. We actually made that warning in the README. The reason for that was basically that in the command line, for people that are, for example, testing or doing benchmark with other applications, we want to make sure that they can call as much variation as possible, and they use the filters depending on their application. So they change a little bit, but the difference is not too big. Now, to the question if the parameters are optimized for plants or they are generic, they are generic, in part because plants are actually very different from each other, and different species behave in a very different way. So I don't see a way to make something particular for plants. So in general, the parameters are generic for basically any organism. And then to the question, are there ways to estimate heterozygosity rate of an unknown plant? So I think the statistics are pretty helpful for that. So you can run the pipeline, you can get the combined DCF, and you can get a population DCF, and then our statistics, our general statistics that we will show later, and our diversity statistics, I think, are very helpful for heterozygous. Depending on the number of heterozygotes, the percentage of heterozygotes, you can see how much heterozygotes. Daniel is telling us that Daniel has a lot of inside information, because he is one of the persons working with that help us to work with NGSEP, and saying that it's important to the GQ used by other tools. So GQ, for people that are not familiar with BCF, is the main piece of information that you get for each genotype that tells the genotype quality. So in general, on BCF files, no matter which tool you use to produce them, it's important to understand that every data point that you genotype comes with a quality score, and that quality score, you can use filters on quality score either at this stage or later after the complete BCF is assembled. You can filter by quality score, by GQ, and that way you can tune. If you can decide if you want to have more genotype calls with more errors, or less genotype calls with a lower error rate. Okay. So any more questions before? Good. So let's keep going with the match variance.
Okay, so that was the first step of the pipeline. Now we have discovered all the variation present in our samples. The output must be something like the one present in these folders. So I will have at the end two files. One of them containing the structural variation detected for the sample, and the other one the single nucleotide variation or small indels. So in the GFF file, I am going to find a list of all the variants, either if they are copy number variants or repetitive regions, or for example, deletions like here, or inversions or insertions. Everything will be reported here, and the good thing about this format is that you can load it in any genome browser and observe these variants compared to your reference genome. And the BCF format will show us for our sample, which is here, what is the genotype that it has at each of the positions. So here are the list of the positions, the reference allele, the alternative allele, and what genotype our sample has. Okay, and that's for both of our samples. This is the ER7A sample, which is basically very similar, but it was genotyped at different positions because it has variation that is different from the other sample. So the first thing to do now is to merge, to obtain a list of all the variant positions of that reference genome so that we can genotype our samples in those positions. So for that, I will use this app, Merge Variants. This app takes as an input a list of all the sequences present in the reference genome. For that, I have this file called secnames, and a list of the VCFs that I already produced. So for that, I will use the sample files. The VCF for the first sample and the VCF for the second sample. And that's it. This should take 1 minute or so, and then after it is finished, I can open the variance detector again to genotype my samples. And to do that, well, first of all, I'll change the name here to genotyping of CBS4C. I will load my sample file, the alignment file again. And the reference genome. Okay, and now here in the known variants file, I will load what came out from the merge variants procedure, which is here. And I will skip all of the structural variation analysis just to genotype these positions. It's also a good idea to lower this threshold so all of the evidence will be output. And that's it. I'll launch the analysis. And now I have to do the exact same procedure for the other sample. I will load here the alignments file. And the reference genome in FASTA format. And the list of variants that was produced. Here I'll check to skip all of the structural variance analysis and lower this threshold. And that's it. Okay, so the list of variant position, NGSEP outputs that as a VCF. So what we get is a clear VCF of each of the positions that were found to be variant in any of our samples. with no genotype or sample information. So it's just a list of positions for the whole genome. And when we finish with the analysis of the genotyping of the samples, I will have two directories, two folders with the genotyping of both samples, like here, and only with the VCF, because I skipped all of the structural variant analysis, so I won't have that file here. This VCF now is going to have information for the variation in the sample and also for positions that are the same as the reference. For example, here, the genotype is exactly the same as the reference. So now that I know all of the positions all of the genotypes at those positions for both of my samples, I want to merge both of these files in one single file in a single VCF. And for that, I have here this app, which is called NGSEP Merge VCF, which takes as input also a list of the sequence names from the reference genome and the output files from the genotyping process. One for each sample. Okay, and that's it. At the end of this process, I will have this directory with a single VCF containing both of my samples. This will look something like this. It's a VCF with all of the positions that are variant with the reference allele, the alternative allele, and the genotype for my samples at each of those positions. And if I have more samples, then they will look like columns here to the right. Okay, and finally, if I have the information of the annotated genome, like in this case, because Saccharomyces is a very well-studied organism, I have it here in GFF format, which are the normal annotation files for most of the organisms, and they are present, for example, in Phytosome. You can download it from there. I can go to the functional annotation app and load these files, the VCF with all my population. That is here, and the GFF file with the annotation. Okay.
Okay, well, seems like there is no demo that is 100% perfect. In the meantime, while the GFF is loading, let's take some questions from the audience. So let's let me go up because we have a question before this too. So, what is it? Good. So, but Alex Hall was telling us, I'm noticing that you're working with G's in the example. It should offer animal genome with the same kinds of input data, right? Yes, the answer is yes. We use a lot G's because, again, it has a small genome. It's easy to work with. It's the same as, because I mean, many people work with G's in the lab, and that's because it's easy to operate. In bioinformatics, it's interestingly the same. So we have a small genome, so data is not so big. So for examples and even for tool developing is good. But definitely the idea is that this is a generic tool that you can use in animals and also in plants. Carol is telling us that she noticed that when the stage you put 0 as the quality score, why? Shouldn't be 40 or 30, something like that. So in the discovery step, we kept a high quality score, 40, which is something that we consider a high quality score. You can even go higher, and that's a good idea because you want to make sure that the deposition is variable. You have a good evidence to know that the deposition is variable in your genome. In the genotyping step, we lower the quality to 0. We normally you can keep a high quality also for the genotyping step already, and then your VCF will only contain higher quality calls. And then we normally choose to lower to zero the genotype, the minimum genotype probability, so that we get in the final VCF we get genotype calls for all the positions, and then with the filter you can filter back by quality if you want. You can take different decisions at the quality step, but then the final VCF will contain all the information. You can pretty much forget that you made that this came from sequencing reads. You can forget all the secondary analysis, and you can take different decisions based on the final VCF. That's the reason mainly why we decide to lower to zero the quality score in the genotyping step. So Alex is telling us, I think that's because there is no analysis being done, and still he wants to map the discovery variance to the reference. That's because there is no analysis being done. Okay, I think he was trying to answer Carolina. You can do all this in such a short time, even with the TV time skipping with the analysis steps. Well, thanks. Yeah, in part of that because again, we use GIS to make the analysis faster. But I think we are doing also a lot of effort to make the software too efficient, so that you can spend. I mean, the processes take a reasonable time, and you can process a whole population in a decent time frame. And also with not that much disk space, we try to save a lot of disk space, and also to be able to make it in memory, that make it efficient memory consumption. Finally, can you please tell us again from where you can access the GFF files? Can we use GenBank for annotation? Unfortunately, no. So the official format that we have for the format that we support for annotations is GFF. GFF is available for plants, for most of the plants. If you download the reference genome from Python, you can use. They also have the GFF that is attached with that reference genome. I think in humans, if you look at GUCSC, you should be able to find the GFF also for human samples. For GIS, is available at the Saccharomyces cerevisiae database. If you definitely have it in another format, there could be a way more or less easy to convert it to GFF.
All right, so I'll show you a couple other things you can do with NGSEP. You can annotate NGSEP using the functional annotation app. I will launch this here with the GFF annotation and the FASTA reference. You can also filter VCF files using the Filter VCF app. You can filter them by GC content to get only the SNPs that are surrounded by a given GC content. You can filter by gene name or by functional role of the variant. You can select or filter out some samples, and you can perform also genotype quality score filters, minimum distance between variants, or keep only the allelic SNPs or invariant sites. So there are a long list of things you can do with a VCF using this filter VCF. And once you have that, you would like to export your VCF to other formats to use it in other applications for downstream analysis. So we have here this app to convert VCFs to other formats like FASTA, structure, HubMap, or Flapjack, or for JoinMap, for example, which needs also some extra parameters. And you can also have some diversity statistics or summary statistics from a VCF. So if you load here your VCF and set the number of samples genotyped, which in this case would be 1 or 2, you can get a good summary of how your VCF looks like. So it is a text file like this. I will just show a little bit. This is from another population, a bean population with 31 variants, and here it has information about the missing data points and also about the annotation, how many SNPs are synonymous, how many SNPs are missense, how many are non-reference. And this is all information you can obtain from a VCF using the apps of NGSEP present in iPlant.
Good.
Okay, thank you very much.
Thanks, Juan. So I'll take a couple of additional questions that we have. This is a very important question. Okay. So we're not sharing anymore, right?
No.
Okay, great. So this is an important question. It sounds like this is straightforward for a small sample size, but how a population genetic study with several samples? The answer is in iPlant, this is true. So what we are releasing right now in iPlant is good if you have a moderate set of samples that you can run one at a time. Definitely, this will not be practical for a lot of samples. In the graphical interface and in the command line, we have made in the graphical interface, there is a process, we have functionalities to run in batch a large number of samples, and we have tested, like in the case of GDS, that you have a large number of samples and not too much information for each sample. We have tested with populations of 400, 600 samples, and it's fine. And in the command line, we have a few scripts that we recommend that you can either integrate into a PBS environment, or you can put together to process a large number of samples. I think one of our hopefully next step that we will make soon in the next few months will be to explore ways in iPlant. So for that we'll keep working with the people of iPlant to devise ways to facilitate the process when you have a large number of samples. So definitely that will be something that we want to work on and have in the next few months. So
so Alex Hall asked if
I think Alex was trying to answer the previous question. Yes. So Alex was saying that if you want to use some shell scripts in association with the command line version, that's definitely what we do. And then translation of VCF files, you can do it with the converter, with our converter. So that's what I currently do, but I'm using the snips now and toss out other variants.
Yeah.
It's my interest in this webinar. You can remove other variants basically because the snips are easy to genotype and you get very high quality information, or you can keep the other variants and they will appear in your statistics and you can do downstream analysis also with variants. Caro, have you actualized all the new models in the command line package? Everything is in the latest version. Command line, no matter from where you run it, if you run version 2.1.4, everything is perfectly, you will run exactly the same code, everything is up to date to the latest version. How we can get the amino acid changes. By now, we have the functional consequences in the VCF file. So it's something also that we need to improve. Basically, in the VCF file, you have the type of change, if it's synonymous, non-synonymous. You have the gene where it happens, and you have the codon position where it happens, where the change happens. Right now, it's only on the VCF, but we will work on a report, or we have options that we are exploring to make that to transfer that information also to a format that is more easy to process.
Where do we get GFF files?
Yeah. And then Paul, I apologize, but missed your answer. How did you generate the GFF files? So if it's the GFF files, so there we have two types of GFF files here. No? One is the GFF file that is associated with the genome. That file normally comes with the genome when you annotate. Just make sure that it's proper GFF3 format. So the gene is called gene and not, for example, gene something or express gene or something like that, to make sure it works. But normally, whenever they release the genome of the species that you are working, it comes with a GFF file that contains annotations. If you are talking about the GFF files describing the structural variation, those are produced by the software by the variance detector, app that we explained here. Okay, any more questions? Okay, Marta, how are we doing time? Do we have 5 more minutes, or should we close?
Yeah, I think if there are no more questions, I will let everyone know that I will send out a follow-up email later today with links to the slides, the video recording, and please a survey to give us feedback, because we really want these webinars to work for our users, so let us know what you need. Let's see. I think you have, yeah. Oh, you have a couple more slides. Go ahead, Jorge.
Right.
Sorry.
So yeah, just to finish, so the last 5 minutes I will tell you guys a little bit of the experiences that we already have. So I think, yeah, let me move this out of the way. So we have again been working on this project for quite a few years already. Just to let you know, this is just to let you know that this is actually being used by several groups. Definitely, our showcase is work that we finished early this year on rice. So we ran this pipeline on 104 rice varieties, taken sequence at different stages by a few collaborators. And we basically were able, using this pipeline, to release it and to put in context a lot of the knowledge on rice genetics, taking into account that a few, a good proportion of the new samples that were sequenced were elite varieties, commercial varieties that were that people eat in Latin America and in North America. And then because we do try to do what's we do apply breeding here, we explore and we present one of our first results on how to take this information to produce SNPs that can be used as markers for marker-assisted selection, and how this works for grain quality in rice. So if you want to hear if you want to read the complete story, this publication has the details. This was a collaborative work with Zen State, USDA, and CIAT. And then this is a list of a few more people that have been working with NGSEP, because of my previous appointments at UConn and at Leuven. People still use either older versions of NGSEP or even the latest version for human and mouse cancers. And the project where I was working for was, in my case, it was discovery vectors for immunotherapy. Then is here you can find a drawing of a publication of what happened at the end of this project, and what people is doing right now with more and more samples. In GIS, we also have a lot of gene discovery analysis that is done in investigation. And then we have our first two success cases. I call success here people from different groups who use NGSEP, who could use it to analyze the data and to produce the results and have their publications ready. So we have here work from University of Georgia, working in peanuts, with design. We have a recent paper, this just came out from University of Antioquia here in Colombia. They work with human pathogens, Clostridium and Trypanosoma. They do assembly and copy number variation, and they use NGSEP for the analysis that you can find here. Here at CIAT, definitely this is our tool, and we use it with several types of data. We have experience with whole genome sequencing, GBS, rat sequencing. We do diversity, we do markers for breeding, we do several different analyses. And then we also work with other Colombian universities and institutions that in different routes, and with different alternatives and different sequencing strategies. And then that was the people I know, but then there is a lot of people, and really happy to share with you the statistics that we have right now of people around the world that are using this tool, that are downloading our software in several countries. We are close to 1,500 downloads this year, and I think this is a pretty okay number for this application. And I don't really know what they are doing. Perhaps they are just looking at the software and living, or perhaps they are actually. But I think that there's quite a few people that is already consistently downloading and using the software, and I think that's great for us. I will finish here. So the acknowledgements. So I will have to acknowledge like half of here, because we have many people here have helped us a lot with this effort. But I mentioned here a few names from the RISE breeding program, from the bean breeding program, and also from the IT department, the people who have the servers up and have everything set up so that we can make a good job. At this time, I would like to acknowledge a lot of the people of iPlant, who have helped us. Marta has been a really great help to allow us to integrate NGSEP into the iPlant environment and produce and even make the webinar that we are seeing today. There is quite a few other people from iPlant that have made a lot of work to make this happen. The funding for for all this work is from the CGI system, from the CRPs of rice, grain, beans, and fruit and bananas. And this is the people. This is the people who I actually like to acknowledge the most. These are the people that in some one way or another have contributed to the development of NGSEP. And this is a great group of young scientists that are doing, I think, a really great job. And on behalf of all of them, I would like to thank you guys for assisting to this webinar. And yeah, you can have a few questions, or otherwise, thank you for coming to us. So,
are there any last questions? I have not seen any additional questions. So I would like to thank all the participants, as well as Juan and Jorge.